Good afternoon, everyone. Very happy to welcome you into the Illumina Centre today. My name is Jen Stobbs. I'm the Product Marketing Manager for the MiSeq i100 series launch. So I've been working with the European team to make sure that everybody knows about the features of the new instrument, and part of that involves inviting you all here today to share some of the advancements and the innovations that have gone into developing the instrument, but why that matters to you and the work that you do every day. So we are launching two instruments. Hopefully you already are aware of them. We are launching a MiSeq i100 and the MiSeq i100 Plus systems. So on the face of it, they look the same from the outside, but they are slightly different inside in terms of the capacity of sequencing that they can deliver. So we'll dig into a little bit more of the detail around that. But the main messages that we've got is around the simplicity. We've done a lot of work to make sure that the instruments are much more easy to use, user-friendly, but also speed in terms of the number of sequencing runs you can do per week is dramatically improved, and the sequencing time per run is a lot faster than it was previously as well. So the simplicity isn't just about how you use the instrument. It's not just about loading a flow cell. We've streamlined and made things much more simple to use right from the library, basically from the sample stage right through until you've got secondary analysis, or even some of our tertiary analysis tools as well. But not just that, but thinking about how the instrument arrives in your lab. Everything, all of these touch points have been streamlined to make things much simpler. The speed, obviously, as I touched on, there is a wider output now, so that with the Plus system you can do even bigger runs than you could on the previous MiSeq. But the speed, the cycle times have been improved so much, and that's mainly because we've brought in that XLEAP new version of SBS chemistry to the MiSeq range now, and then made even more innovations than we had previously done with our XLEAP chemistry, so that the speed of the instrument is 4 times faster than the previous MiSeq instrument. But that's great if it's simple and faster, but you need high quality data, and that's what Illumina is known for, and why our MiSeq old instrument has been so popular, and it's still by far our most popular instrument. But it's because Illumina delivers really high quality data, and it is well trusted across biological sciences. So we needed to make sure that the sequence and data quality that comes off is as you would expect or better. So just briefly, these are the instrument configurations that we had. So on the left hand side, the MiSeq i100 system, this is the lower throughput option. So it can run the 5 million read and the 25 million read flow cell, which covers basically what the current MiSeq offers. But the instrument that is available now to order now is the MiSeq i100 Plus. So it can do the 5 million and the 25 million read flow cells, but it can also sequence with a 50 million and a 100 million read flow cell, which really helps bridge that gap between the MiSeq range and the NextSeq range, and it opens the discovery power up to more applications. So you can start thinking about things like if you've done a targeted DNA sequencing experiment, maybe you want to have a look at RNA-seq, and you can start thinking about that on the same instrument now, whereas previously you might have had to move up to a NextSeq to do that. So there's a much bigger range in terms of output on the two instruments. So to touch on the simplicity, as I said, we've sort of reimagined this right from setup right through to data analysis. And that setup starts from the minute that the instrument arrives in your lab. So with our older MiSeq and the MiniSeq as well, you previously had to order it through a salesperson, and then one of our engineers or a field scientist would come out and install that instrument for you. This one is so much simpler. You can actually do it yourself if you want to. We already offer this with our iSeq instrument, but the improvements in the software and the way this is set up means that you can get this delivered. White Glove will come and place it on the bench for you, and then you can look after the setup. It's very wizard based. It's a few steps. The only thing that you would need to do is to arrange with your IT to get networking details so that you can network the instrument if you want to. So you can get sequencing started in as little as an hour, assuming that you've already got those IT details. When we ship it, it will come in a cardboard shipper rather than a wooden crate, which is obviously a bit more environmentally friendly and much easier to recycle. And it's a lighter instrument as well. So the old MiSeq was 50 kilograms, whereas the new MiSeq is only 36 kilograms. And it is a much smaller footprint. So it's 54% of the size of the original MiSeq. So it takes up much less, quite a bit smaller size of space as well. So no specialist tools required. White glove will place it on the bench, and then you can work through the wizard-based tool to help get it set up. Saying that, if you weren't confident to do that, we can still arrange to have an engineer come and install it and do some training while they're there as well. So that is also an option. In terms of the whole pipeline, so obviously we've streamlined the setup of the instrument. So now it really is, it's much easier. It's 3 steps that you can do in 20 minutes to get your run set up, ready and going. Everything is labelled on the flow cells now, exactly where you need to place the libraries. You can do the run setup either on the instrument, or you can do it at a desktop computer if it's networked. And that can all be done in advance of the run. Denaturation of the libraries now is also done on board, which saves in those extra steps. And cluster generation, as with all of our newer instruments now as well, cluster generation is done on board the instrument. So there's no additional instrumentation that you need to be able to do cluster generation. And this is all to just reduce the hands-on time. So it's simple to set up the run, and it's simple to do the wet lab work to get it running. With the new MiSeq i100, there is a DRAGEN on board the system. Now, DRAGEN is a secondary analysis software tool that will align your sequencing reads to the reference genome that you provide, and basically it will then create your VCF files. So there are some DRAGEN apps that are available on board the instrument, depending on the workflow that you choose. But you can also access the DRAGEN apps in BaseSpace Sequence Hub, or you can use your own bioinformatics pipelines if you choose to. But that DRAGEN analysis that is on board, there is no additional cost for that. There's no license fees, it's already included in the cost of the instrument. Maintenance is now much simpler. So the way the cartridge and the flow cell is designed is all the fluidics are within the cartridge. Within the flow cell. So there's no fluidics actually inside the instrument other than to take the waste into the waste bottle. So that removes the needs for doing washes between runs. So at the moment, to make sure that you don't have contamination between runs, you have to do all of those wash steps to make sure it's all clear and ready to go for the next run. And in terms of disposal of the core consumables as well, you don't need any special tools to take them apart. But it does now separate, it just pops, and you can see that in the demonstration later as well. They pop apart so that you can recycle the plastics, and it's just standard plastics as well, so it's fairly easy to recycle. But something that's quite exciting is the fact that we've removed formamide. So disposal of formamide on the MiSeq was always an issue because it's a nasty chemical. So if we can remove that from the whole experience, then it's best for everyone. So there's no more formamide in the current reagents. We've also streamlined the packaging. So with the original MiSeq system, you had to have polystyrene boxes with dry ice stored at -20 and fridge temperatures as well, but now that packaging has been dramatically reduced, so the volume of core consumables is so much smaller. That's an 85% reduction in packaging materials. So when you think about it in terms of the environmental impact, we need less lorries to get our core consumables to your lab, so you're reducing your carbon impact in that sense as well. As I say, you can reduce your waste by recyclable plastics, and we don't need to use ice packs and dry ice when we're shipping these consumables as well. And we've extended that into the lab. So with the XLEAP chemistry developments that we announced for the NovaSeq X the NovaSeq X series, we did announce ambient shipping, which was a really big advantage for a lot of people. But now we've taken that one step further. So when you're storing these reagents in the lab, they no longer need to be in a fridge and a freezer. You can just store them on the shelf, which means that they are ready to go whenever you need them. You don't need to think about thawing them or bringing them back up to room temperature, which is a really big advantage if you need to have a faster turnaround time. It also means you're less likely to have running consistencies because you're not worried about whether you've left it long enough to thaw, which is obviously quite a big one. It makes a big energy impact in terms of storage as well. So if you've got a lot of organisations now have their own carbon reduction targets, so if you're not needing to maintain as much fridge and freezer storage, that is a big advantage for everyone. In terms of shelf life, so the shelf life of the reagents will be 12 months from manufacture, but 3 months at least guaranteed in your hands. We do plan to extend that. So as time goes by, once we've got 6 months on the clock, then we will do a lot more testing to make sure that those reagents still perform to the same standards after 6 months, and then we will announce like an extended shelf life as well as a guarantee to your hands. But at launch it's 3 months. This just gives you an idea of the interface that you can see, so you can see the run status either on the instrument or on the desktop. You can check in to see how well it's performing. You've got flexible run management, so you can either set up the runs again on the instrument screen, or you can do it at a lab computer that is networked. And you can start planning your runs in advance of having done the lab work so that you know it's ready and set up to go. And DRAGEN reports as well. So because the DRAGEN analysis is now on board, you can actually access that information on the screen on the instrument or on the desktop that's networked as well. So you can access those results straight away. With the DRAGEN analysis, DRAGEN is becoming more of a well-known concept now, but DRAGEN provides advantages because it is a much faster way of doing the secondary analysis compared to the LRM system that we used to use, or things like BWA, GA to K pipelines. DRAGEN performs secondary analysis a lot quicker, so you get to your answers quicker. There will be onboard analysis for things like library QC, small whole genome sequencing, and we've got infectious disease and microbiology panels as well. But you can also access everything that's on BaseSpace Sequence Hub in the cloud, so there are additional DRAGEN apps. And you can also use the BCL Convert and then run your traditional bioinformatics pipelines as well. It's incredibly efficient using DRAGEN, so it's also set up, because it's set up as part of the run setup, it's defined at the beginning stage, it automatically happens. So as soon as sequencing is finished, the DRAGEN analysis begins, so there's no user touch points, it's drastically reduced. These are some examples of the ones that are available on board. So you've got the DRAGEN BCL Convert, which takes the base call files and turns them into FASTQ files. The vast majority of bioinformatics pipelines use that process so that you can then take the FASTQ files and do anything that you want to do with them if you have alternative pipelines. We've got library QC that helps you analyse the performance of the run, DRAGEN small whole genome sequencing, and DRAGEN microbial enrichment plus. We do have more workflows that are supported on the instrument, so we're mostly covering the most popular applications that MiSeq and our low throughput instruments are used for. So that is mostly oncology small panels, but also infectious disease and microbiology studies as well. So there are 18 supported workflows with 10 different applications available at launch. We will add to that, and that's not to say that you can't do other applications on the MiSeq as well. It's just that we might not have generated the internal data. So if you do need to troubleshoot, we might not have the data internally to be able to help as much, but we will be adding to that over time. So these are some of the supported applications we've got based on Illumina's library preps that we already have available. So in the infectious disease and microbiology space, we've got some of the respiratory pathogen panels, and then some of the microbiology applications as well. And we've got those cloud and onboard DRAGEN analysis options for you to choose from, whichever is most suitable for your pipelines. So now we'll touch about some of the innovations that have helped us speed the process up, what makes it so much faster. And a lot of it comes from the XLEAP chemistry innovations. But it does now mean that if you're doing small whole genome sequencing on a 2x300 run, where it would have previously taken 56 hours on the older MiSeq, you're now down to 15 hours. So you can do it overnight instead of it taking all week, which is a big advantage in terms of return on investment for the instrument, because you can do more runs in the same time period. And because of the expanded output, because if with the MiSeq i100 Plus system, you can do 50 and 100 million reads, you can now expand the types of research that you do with this instrument. So traditionally, MiSeq has been great for things like 16S amplicon sequencing. You can still do that on the MiSeq i100, but you could potentially think about doing things bigger. So you might want to switch to shotgun metagenomics, or you might want to think about transcriptome profiling and looking at RNA-seq. And now you can do that all on the one instrument. This just gives you an idea of the sample numbers that you can do. So small whole genome sequencing, for example, on a 5 million read flow cell, you might get 10 samples on it. Whereas if you want to do a larger experiment on the MiSeq i100 Plus 100 million read cartridge, you could do 200 samples. So you've got a really big range of flexibility in there. Comparing output from the legacy MiSeq system against the i100 series. So the output is different. So you can now go from, the lowest is a 1 gigabase, right up to 30 gigabases. And the read numbers is now 5 to 100 million reads. We don't have a flow cell option for the 1 million read, which we do have on the iSeq and the MiSeq. But in terms of popularity of flow cells, the 1 million read flow cell was quite often used for QC, but a lot of people use the bigger output flow cells, so that's why we've stuck to between 5 and 100 million. And the runtime is a big factor, so reducing it, you can get your sequencing results in as little as 4 hours, but the maximum one is 15 hours anyway, which is a huge improvement on what it was previously 56. So with the new system, we've got 4 times the output and 4 times faster run times, which is a big game changer for a lot of labs. And one little technical advantage as well, which I think is very interesting, is the new instruments run with index-first sequencing. Now this means that it sequences the first index on read 1, it sequences the second index, and then it goes back to do the sequencing of read 1 and then the sequencing of read 2. You can choose to do it the old way if you want to. There is an option to go back and do that. But index first might open up some new applications. So if you are doing things like QC runs, where you might be doing it for a run on a NovaSeq X, you want to check that your libraries are balanced, you can get that information really, really quickly, because you can see how balanced the indices are in a couple of hours, rather than having to wait for the whole thing to finish. And then you can still get all of the read information if you want to as well. But there are applications that this might lend itself to, some newly designed library preps, for example, that might help you attach the indices in a way that means that you can detect pathogens immediately, rather than having to wait for all of the data to come back. So it'll be interesting to see how that's applied in real life. And finally, touching on the quality of the data. So it's great that we've simplified it and made it faster, but we can't sacrifice quality, and we haven't. It is actually considerably better with the MiSeq i100 series as well. And this comes with those XLEAP chemistry improvements. So it's a novel polymerase. I can't share any details about what that is because obviously it's top secret. But there is a lot more developments in this version of XLEAP chemistry compared to what we've released on the NovaSeq X and what is available on the XLEAP 1K, 2K. It is a slightly different chemistry. It's still a blue-green dye, but it is different to what is on the NovaSeq X. And there's a lot more advantages in terms of the stability of the reagents. So more of those enzymes that are in there are now lyophilized, and there are technological advancements to how those are stored, and that gives this a much higher level of stability, and that's what allows us to offer ambient shipping and storage, so it's some huge advantages there. It also enables faster cycle times, so actually being able to do each cycle is much quicker. Obviously that reduces the overall sequence and run time. It's improved the accuracy a lot, which will show you some data in a moment. And it's also more cost effective. So the new flow cells are patterned flow cells rather than random, which means that we can miniaturize the chemistry by a lot. And also because the chemistry is much more stable, it means we can pass those cost savings onto the list price. So on average, across all of the flow cells, it's 40% cheaper compared to previous MiSeq reagents. So not only is it faster and easier to use, it's cheaper to use as well. So you can potentially expand the discovery power, the different types of research that you do on the instrument in a much more cost-effective way. This shows us the quality improvements. So on the left-hand side, this shows us the error rate, comparing the old MiSeq in grey with the MiSeq i100 in orange. And as you can see, the error rate is dramatically reduced, particularly for read 2. And on the right-hand side, you've got the Q30 score, which is, again, improved, but even more so for the read 2. So we're comparing the old MiSeq with the new MiSeq, and the Q30 percentage there is considerably better, which is demonstrated on this one. And this is my favourite demonstration of the quality of the new chemistry. So in grey, you can see how over the cycle numbers, the quality drops off. And the Q30 score drops quite considerably, particularly when you get to read 2. But in blue, you can see the quality score, the Q30 score, with the MiSeq i100 series. And it barely drops off. So you can see on the table there, for the read 2, the last 10 cycles, the average Q30 score is improved from 10.7% to 91%, which is a dramatic improvement. And does mean that you can then potentially stick with doing those long reads, which is what MiSeq has always been so popular for, doing the 2x300 runs. And Illumina offer a huge amount of support. So MiSeq has been by far one of our most popular instruments. Because it's such a flexible instrument and it's been out there for quite a long time now, we do have over 10,000 MiSeqs globally. In different labs, but it has been cited in publications over 160,000 times. So it's a very, very trusted chemistry. So we have developed that, take all the best bits, develop it and make it easier and faster to use. And supported is our global service and support team. So we have a huge number of field service engineers and field application scientists, technical application scientists who are there on hand to help you if you have any troubles or if you want to think about different workflows, how you expand your research. We've got a great support network to help you do that. This one is a nice example of a comparison of a 16S metagenomics run between MiSeq and MiSeq i100. So as you can see, because 16S metagenomics is a really low diversity run, you tended to need quite a lot of Phi-X spiked in, and it usually was around 20%, depending on how many samples you've got in the run and the diversity. But now on the MiSeq i100, what you can see on screen is with only a 5% Phi-X spike in. So it tolerates low diversity runs in a much better way. The error rate is reduced as well. It's only 0.5%, so it's a much better sequence and quality run. And I'm going to stop there because we do have some sessions on more specifically on the workflows and the applications